Tantor Audio, a division of Recorded Books, presents Cannabinoids and the Brain by Linda A. Parker. Narrated by Erin Deward. Preface. My keen interest in the endocannabinoid system began with an airmail envelope from Israel that arrived in my faculty mailbox in the year 2001. In late 1999, Cheryl Limebeer and I published a paper showing that THC reduced nausea in a rat model that we had had a role in developing. Shortly after the paper was published, I received a letter from Professor Raphael Mechoulam suggesting that we test non-psychotropic cannabidiol in that model. I was thrilled. The letter began a very exciting and fruitful collaboration that brought me into the exciting world of endocannabinoids. In 2002, at Professor Mechoulam's kind invitation, I attended my first meeting of the International Cannabinoid Research Society in Monterey, California, at which I met Professor Mechoulam and several other outstanding researchers who would later become collaborators. In addition to Professor Mechoulam, our group has had the good fortune of collaborating with such leaders in their field as Roger Pertwee, University of Aberdeen, Keith Sharkey, University of Calgary, Danielle Piomelli, University of California at Irvine, Aaron Lichtman, Virginia Commonwealth University, Benjamin Kravitz, Scripps Research Institute, Lauren Parsons, Scripps Research Institute, Alexandros Makrianis, Northeastern University, Paul Fletcher, Center for Addiction and Mental Health, Brian Kolb, University of Lethbridge, Klaus Peter Ossenkop, University of Western Ontario, John Salamone, University of Connecticut, Ganesh Thames, Northeastern University, and Matias Lopez, University of Oviedo. And with my wonderful colleagues at the University of Guelph, Elena Coloris, Francesco Leri, Boyer Winters, Bettina Kalish, and Craig Bailey. Over the past 10 years, these collaborations have provided outstanding training opportunities for members of my laboratory, including Cheryl Limebeer, Aaron Rock, Martin Sticht, Kiri Wills, Leslie O'Brien, Shadna Rana, Catherine Turk, and Gavin Petrie. It is these hard-working, enthusiastic, highly skilled people who keep the process of discovery going in the laboratory. Nothing would happen without them. This book is an expansion and update of a review paper which Raphael Mechoulam kindly invited me to co-author that appeared in the Annual Review of Psychology. Our collaborations over the past 15 years have always taken my research to new vistas. Astute and careful editing by Paul Bitka at the MIT Press has made the book a much better read. I thank him for his contributions to the clarity of my prose. I draw the reader's attention to 4 outstanding edited volumes on cannabinoids and endocannabinoids that have been published in the past few years. Handbook of Cannabis, edited by R.G. Pertwee. Handbook of Experimental Pharmacology, Volume 231. Endocannabinoids, edited by R.G. Pertwee. International Review of Neurobiology, Volume 125. Endocannabinoids, edited by L. Parsons and M. Hill. And Cannabinoids, edited by V. D. Marzo. For more information, see the information for healthcare professionals on the Health Canada website at hcdhc.infopros-eng.php. The National Institutes of Health recently prepared a videocast of a 2-day conference, Marijuana and Cannabinoids, a neuroscience research summit, held in March of 2016, and featuring presentations by some of the foremost NIH-funded researchers. I encourage readers of this book to find a free evening, open the link, videocast.nih.gov, and hear about the current state of the research in this exciting field. I also recommend The Scientist, a video documentary produced by Fundación Cana and available on YouTube, that provides a glimpse into the scientific and personal life of Raphael Mechoulam, the father of the endocannabinoid system, a humble, brilliant, generous, and truly delightful human being. I have had the great fortune of being able to call Professor Mechoulam my friend. 1. An Introduction to Cannabis. The cannabis plant has been used for recreational and medicinal purposes for more than 4,000 years, but only recently has scientific investigation of its effects begun to be fruitful. In 1964, at the Hebrew University of Jerusalem, the chemists Yael Gaoni and Raphael Mechoulam discovered the principal psychoactive ingredient in cannabis, delta-9 tetrahydocannbinol, often referred to as THC. This discovery finally provided the scientific community with the tools needed to investigate the properties of this plant. Although it was clear that the psychoactive effects of THC were produced by its action on the brain, exactly how that happened was not discovered until 20 years later, when a Lynn Howlett's group at the St. Louis University Medical School discovered the cannabinoid 1, CB1, receptors. The brain distribution of these newly discovered receptors was found to be consistent with the known pharmacological effects of THC. This discovery raised a question. Why would the brain have receptors for a material derived from a plant? Obviously, the brain must produce chemicals similar to THC. Shortly thereafter, Mechoulam's group discovered anandamide, AEA, and 2-arachidonyl glycerol, 2-AG, which are called endocannabinoids because they act on the same receptors as THC. And a second cannabinoid receptor was discovered, CB2, found primarily in the immune system. The discovery of the site of action of THC has uncovered a new brain chemical system that is intimately involved in all aspects of brain functioning. Since the aforementioned discoveries, a large number of investigators have provided considerable scientific knowledge about the metabolism, biochemistry, and pharmacology of THC, mostly in animal models. In a recent comprehensive review, Patcher and Kunosh state that modulating endocannabinoid activity may have therapeutic potential in almost all diseases affecting humans, including obesity/metabolic syndrome, diabetes and diabetic complications, neurodegenerative, inflammatory, cardiovascular, liver, gastrointestinal, skin diseases, pain, psychiatric disorders, cachexia, cancer, chemotherapy-induced nausea and vomiting, among others. This bold statement is based on considerable preclinical scientific evidence in each of these fields. This newly discovered system is of great importance and can explain many of the medicinal claims that have been made for medical marijuana. However, because of legal constraints, very little clinical work has been done in humans. In modern medicine, there are few approved conditions for the use of cannabis and cannabinoid-derived medicines. This is due to the rather limited, high-quality human clinical trials with cannabinoids. To be approved for use in a patient population, a drug must undergo clinical trials. Clinical trials have 3 phases to assess a new medicine for safety and effectiveness. Phase 1 tests for safety. A small number of people are given a small dose of the drug under careful supervision. This is not done to test whether the drug works for the condition under review. It is done to test for side effects. In Phase 2, the drug is given to people who have a medical condition to determine whether or not it helps them. Phase 3 trials are conducted only for medicines that have already passed the first 2 phases. In Phase 3, the drug is compared against existing treatments and/or placebo, using randomized assignment to treatment groups, randomized control trials, abbreviated RCT, and double-blind procedures. Neither participant nor the researcher knows to which group a participant was assigned. These clinical trials often last a year or longer, involving several thousand patients and often millions of dollars. According to a recent systematic review and meta-analysis of evidence, a total of 79 RCTs of cannabinoids as medicine for nausea/vomiting, appetite stimulation, chronic pain, spasticity in multiple sclerosis, MS, depression, anxiety disorder, sleep disorder, psychosis, glaucoma, or Tourette syndrome, most studies suggested that cannabinoids were associated with improvements in symptoms. However, when the quality of each study was assessed using the Cochrane Risk of Bias tool, which grades the rigor of the design of a clinical trial, there was moderate quality evidence to support the use of cannabinoids in the treatment of chronic neuropathic or cancer pain and spasticity due to MS. There was low-quality evidence suggesting that cannabinoids were associated with improvements in nausea and vomiting due to chemotherapy, weight gain in HIV infection, sleep disorders, and Tourette syndrome, and very low-quality evidence for an improvement in anxiety. A number of methodological weaknesses were identified in these studies, including failure to appropriately handle participants who withdrew from the trial, selective outcome reporting, inadequate description of methods, inadequate randomization, and inadequate blinding. An additional limitation was the very small sample size of most of the RCTs, which means that they lacked the power to detect any difference in the treatments. Multiple different cannabinoids and routes of administration also were evaluated in various studies. In view of these limitations, Whiting et al. Urged that large, robust RCTs be conducted to confirm the potential medicinal effects of cannabinoids. However, such trials are expensive and are difficult to perform with substances that are highly regulated. Therefore, most of the evidence of the potential medicinal promise of cannabinoids is based on experimental preclinical studies with animals in which greater scientific rigor can be applied to the design of a study. The numbers of such studies have increased exponentially since the discovery of the endocannabinoid system and the mechanism of action of cannabinoids in the 1990s. Results clearly demonstrate the promise of cannabinoids for treatment of several disorders of the nervous system, including anxiety, post-traumatic stress disorder, PTSD, chronic pain, spasticity in MS, epilepsy, obesity, nausea, and vomiting. A brief history of cannabis use. Early in agricultural history, more than 10,000 years ago, cannabis was grown along the rich banks of great rivers, including the Huanghe in China, the Indus in India, the Tigris and Euphrates in Mesopotamia, and the Nile in Egypt, for the purpose of producing hemp fibers. The oldest known written record of cannabis use comes from the Chinese Emperor Shen Nung, who in 2727 BC investigated botanical compounds for the potential medicinal properties, using himself as a test subject. As the father of Chinese medicine, Shen Nung is said to have written the original Pencao, listing hundreds of drugs derived from vegetable, animal, and mineral sources. However, no original text exists. The oldest pensao was compiled in the 1st century A.D. by an unknown author who claimed to have incorporated the original compendium into his book, which contains a reference to ma, the Chinese word for cannabis. In the 2nd century A.D., the Chinese surgeon Hua Tuo used a mixture of cannabis resin and wine as an anesthetic to perform painless surgery. The Chinese were also the first people to report the use of recreational cannabis. India was the first culture to use cannabis for religious and social purposes. In the 4 holy books, known as the Vedas, the god Shiva became angry after a family squabble and takes refuge in the cool shade of a cannabis plant. He eats some of the plant, and it becomes his favorite food. Shiva became known as the Lord of Bhang. Bhang is not merely the plant, but also a liquid refreshment made with its leaves. Ganja and charas, too, were made from cannabis in India. Ganja, made from the flowers, is more potent than bhang. Charas is made from the resin of the flowers and is much more potent than ganja. It is similar to hashish. Bhang is used in a manner similar to alcohol in the West, often in conjunction with weddings and special festivities. According to Indian folk songs dating back to the 12th century, bhang was also consumed by soldiers before they went into battle. At the turn of the 20th century, the Indian Hemp Drug Commission, summoned to investigate the use of cannabis in India, concluded that the plant was an integral part of the culture and religion of the country. Indeed, the commission concluded that, besides a cure for fever, bhang has many medicinal virtues. It cures dysentery and sunstroke, clears phlegm, quickens digestion, sharpens appetite, makes the tongue of the lisper plain, freshens the intellect, and gives alertness to the body and gaiety to the mind. Cannabis was introduced into Western medicine in 1839 by William Brooke O'Shaughnessy, a surgeon who learned of its medicinal benefits while working for the British East Indies Company. It was promoted for analgesic, sedative, anti-inflammatory, antispasmodic, and anticonvulsant properties for which preclinical animal models provide considerable support. In fact, cannabis was said to be the treatment of choice for Queen Victoria's menstrual pain. However, in the 1900s, specific medicines were developed for each of the mentioned indications, and medicinal use of cannabis declined in Western countries. In the middle of the 19th century, Charles Baudelaire, Théophile Gautier, and Jacques-Joseph Moreau, all members of the Parisian Club d'Achichon, wrote about the psychological effects of cannabis. In 1845, Moreau, a psychiatrist, published a book titled Hashish and Mental Illness, in which he described psychological phenomena he had observed in experimental participants, including happiness, excitement, dissociation of ideas, distortion of time and space, sensory enhancement, delusions, emotional fluctuations, illusions, and hallucinations. It is presumed that Moreau's participants consumed large amounts of hashish orally. In the United States, the history of the use and the prohibition of cannabis have been clearly described many times. Early in the 20th century, marijuana was used for its psychoactive properties, mostly by Mexicans and African American jazz musicians in a few large cities. In fact, the wave of immigrants who entered the southern U.S. from Mexico in that period first brought the drug under suspicion by authorities and eventually led to its prohibition. Its recreational use first became widespread in New Orleans, then gradually spread to other cities. In a campaign to outlaw the drug waged by the head of the Federal Bureau of Narcotics, Harry Anslinger, it was alleged that marijuana use led to violent crimes. In the late 1930s, newspaper stories in large cities were filled with stories about killer weed. In 1937, the U.S. Congress passed the Marijuana Tax Act, which effectively banned the use of the drug in medicine and outlawed it as a dangerous narcotic, effectively terminating research on it for about 25 years. International control of cannabis. Late in the 19th century, the British Parliament required the colonial government of India to establish a Royal Commission on Opium and an Indian Hemp Drugs Commission. The latter concluded that moderate use of cannabis had no appreciable physical effects on the body, no harmful effects on the brain, and no adverse effects on morality. It recommended a system of taxation, control, and restriction, but not prohibition. Despite the reports, use of cannabis as a medicine faded as improved technology facilitated the study of pharmacology and organic chemistry, the isolation of active ingredients from natural products, and the synthesis of new molecules. Morphine was identified and isolated in 1805 by Friedrich Serturner, and the subsequent development of the hypodermic needle provided a means of administering it for rapid pain relief. Later in the 19th century, aspirin and other medications, among them barbiturates, were developed. Complex, unrefined herbal medicines became of less interest to mainstream medicine. When efforts to control the use of cannabis arose in Europe and North America in the middle of the 20th century, the medical community did not defend its use as a medicine. Early in the 20th century, there was a growing interest in international cooperation in the control of opium. During the Second Opium Conference, held in Geneva in 1925, cannabis and the resin used to produce hashish were also discussed. In 1954, the World Health Organization reported that cannabis had few medical benefits. In 1961, an international treaty called the Single Convention on Narcotic Drugs established limits on the production, manufacture, and extraction of raw materials from what were determined to be narcotic drugs. Psychoactive substances, drugs, were placed in 4 schedules, the most restrictive of which contained drugs viewed to be particularly dangerous for abuse liability and to have little therapeutic value. The cannabis plant, its resin, its extracts, and its tinctures were placed in the most restrictive schedule. Subsequently, at the 1971 UN Convention on Psychotropic Substances, THC and its isomers were placed in Schedule 1, which prohibited all use except for scientific purposes and very limited medical purposes by duly authorized persons specifically approved by a governmental body. Other cannabinoids, among them cannabidiol, were not controlled under the 1971 UN Psychotropic Convention. These non-psychoactive cannabinoids are still not controlled by many countries, e.g. Great Britain, but the United States and Canada have chosen to include them in the same restrictive schedule as THC. For an excellent discussion of how various nations around the world have interpreted and implemented the UN Treaty and have devised regulatory tools to distinguish between medical and non-medical use, see Mead, 2014. Constituents of cannabis. Although more than 100 plant cannabinoids, phytocannabinoids, have been identified in the cannabis plant, there's essentially only one compound that causes the typical marijuana effects, THC. However, other compounds, including cannabidiol, abbreviated CBD, can modify the effects of THC. The sought-after euphoria produced by smoking marijuana is entirely attributable to THC and not at all attributable to the other cannabinoids found in the plant. THC is not evenly distributed throughout the plant. It is absent from the roots and the seeds, and is found only at very low concentrations in the stems. The lower leaves contain less THC than the upper leaves, typically 2 to 3%. However, unpollinated all-female floral material called sinsemilla, meaning without seeds in Spanish, is a primary source of THC, with concentrations as high as 25%, and other cannabinoids. However, the concentration of cannabinoids within individual plants and tissues is due mainly to the presence or absence of glandular or capitate trichomes, small structures in which cannabinoids are synthesized and sequestered. Varieties of cannabis plants differ in their cannabinoid contents. The concentration of THC in industrial hemp is less than 0.3%. In hashish in the 1960s, it was about 5%. Whereas in marijuana it was about 2 to 3%. In the 21st century, strains of marijuana have been developed that contain up to 25% THC. Because all its types are able to interbreed, many academic botanists argue that cannabis is one polymorphic species, and that the biochemical cannabinoid and terpenoid profiles of different strains of cannabis account for their differing effects. On the other hand, there is recent genetic evidence indicating that hemp is genetically more similar to cannabis indica than to cannabis sativa. The cannabinoids identified from cannabis are of 11 types: delta-9 THC, delta-8 THC, cannabidiol, CBD, cannabigerol, CBG, cannabichromene, CBC, cannabinoidil, CBND, cannabialsoin, CBE, cannabicyclol, CBL, cannabinol, CBN, cannabitriol, CBT, and miscellaneous cannabinoids. In addition to cannabinoids, the cannabis plant contains hundreds of non-cannabinoid constituents, including terpenoids, which give the plant its characteristic smell and which are also found in hops, a close relative to cannabis. In vitro and in vivo, some terpenoids have been found to have anti-inflammatory, antibacterial, and anti-anxiety effects. However, no clinical trials support those claims. Various combinations of cannabinoid constituents and their interaction with terpenoids could provide diverse therapeutic results, accounting for why people claim different symptomatic relief from using different strains. However, all the evidence is anecdotal. Human clinical trials will be needed to harvest the treasure trove of compounds found in the cannabis plant. THC-type cannabinoids. Of the various components, THC types have received the most scientific investigation. The effects of delta-9 THC, commonly called simply THC, will be highlighted throughout this book because it is the primary psychoactive cannabinoid found in cannabis. Very small quantities of delta-8 THC are also found in cannabis. Meshulam et al. Isolated numerous cannabinoids from Lebanese hashish that had been confiscated by Israeli police in Jerusalem in the 1960s and elucidated their structures. Those compounds were then tested for cannabinoid activity in monkeys, and only delta-9 THC was found to produce the typical psychoactive effects of marijuana. Once delta-9 THC had been isolated, Roger Pertwee developed the ring test, a quantitative in vivo assay for psychotropic cannabinoids based on how long a mouse placed across a horizontal ring remains immobile or cataleptic. Later, Billy Martin et al. Used the ring test and 3 other bioassays in what came to be known as the mouse tetrad assay. It included catalepsy, hypokinesia, inactivity, hypothermia, reduced body temperature, and antinociception, pain relief. The mouse tetrad assay is used to screen for psychotropic cannabinoids, all of which, in contrast to many other classes of drugs, generally show similar potency in all 4 of these behavioral measures. After the identification of THC, several investigations on humans showed that when THC was taken orally or intravenously, or when it was inhaled in smoke, it showed substantial potency similar to that reported by recreational cannabis users. At present, 2 analogs of THC that have been approved by the U.S. Food and Drug Administration are available in the United States in capsule.
Sample complete. Ready to continue?